And now to continue with the rest of our presentation, we will be going on towards Down syndrome. So Down syndrome is a genetic condition that affects approximately 1 in every 700 births worldwide. And as you can see in this image, this man has unfortunately confused it with post-traumatic stress disorder. He has created an abomination of a disorder called post-traumatic Down syndrome. How unfortunate. And you can see in these pictures some common characteristics of Down syndrome. So not only does it affect physically, it also can cause mental disabilities and difficulties. So here are some facial features that are common, such as upturned ears, a flat nose bridge, a short neck, and such.
To understand how Down syndrome develops, we need to first understand chromosomes. And chromosomes are structures in the cells of our bodies that carry genetic informations. And we typically have 23 pairs of chromosomes for a total of 46 chromosomes in us human beings. For other species, they have different amounts of chromosomes and different amounts of chromosome pairs. And this is a distinctive feature of a species, is the number of chromosomes they have.
So trisomy 21. Down syndrome is caused by a genetic condition called trisomy 21. Trisomy means there are 3 copies of a particular chromosome instead of the usual 2 copies. In Down syndrome, there are 3 copies of chromosome 23. And this is an image of a karyograph of someone who has Down syndrome. As you can see, in the 21st, there are 3 21st chromosomes.
So, on the topic of non-disjunction events, trisomy 21 occurs due to a problem called a non-disjunction during cell division. Normally, during cell division, chromosomes are separated into 2 equal groups and are distributed into new cells, and this is in meiosis. In a non-disjunction event, the chromosomes fail to separate properly, resulting in one cell having an extra chromosome and the other cell having one fewer chromosome.
So in Down syndrome, non-disjunction occurs in either one of the parent's gametes, so either the sperm or the egg prior to fertilization. When the gamete with an extra copy of chromosome 21 combines with the other parent's gamete during fertilization, the resulting zygote has 3 copies of chromosome 21. This extra chromosome causes the physical and developmental characteristics of Down syndrome.
There are 3 types of Down syndrome, actually. Trisomy 21, the most common type, mosaic Down syndrome, and translocation Down syndrome. Mosaic Down syndrome and translocation Down syndrome occur due to different genetic mechanisms and are less common than trisomy 1, which is a result of a non-disjunction event during meiosis that results in an extra copy of the 21st chromosome.
So, in conclusion, Down syndrome develops due to the genetic condition called trisomy 21, which occurs when there are 3 copies of chromosome 21 instead of the usual 2. This extra chromosome is caused by non-disjunction during cell division in one of the parent's gametes before fertilization. Understanding how Down syndrome develops is important for developing effective treatment and support for individuals with the condition.
Down syndrome can actually be detected prenatally through a variety of tests. And some of the common methods of testing are, for example, screening tests. Non-invasive prenatal screening, NIPS, is a blood test that analyzes fetal DNA in the mother's blood to detect any chromosomal abnormalities, including Down syndrome. It is usually done after 10 weeks of pregnancy and can be combined with first trimester screening. And combined first trimester screening is a test that combines a blood test and an ultrasound to estimate the likelihood of a fetus having Down syndrome. And it is typically done between 11 weeks and 14 weeks of pregnancy. And a second trimester maternal serum screening is a blood test that measures the levels of certain substances in the mother's blood to determine the likelihood of the fetus having Down syndrome, and it is typically done between 15 and 20 weeks of pregnancy. In addition to screening tests, there's actually diagnostic tests, which we'll be getting into now.
There are also diagnostic tests such as chronic villus sampling, short form SVS. This is a procedure that involves taking a small sample of the placenta to analyze the fetal chromosomes. It can be done between 10 and 13 weeks of pregnancy, but it's also important to mention that there is a very small chance of miscarriages as it can directly harm the fetus, but it's also very rare, so don't be afraid. And yeah. And then there's also amniocentesis, and this is a procedure that involves taking a small sample of amniotic fluid to analyze the fetal chromosomes. It's typically done between 15 and 20 weeks of pregnancy. It is important to note that these tests carry a small risk of miscarriage and should only be done if there's a medical need or if the parents have a high risk of having a baby with Down syndrome. Counseling and support should also be provided to help parents make informed decisions about prenatal testing.
So what is karyotyping? Karyotyping is a laboratory technique used to visualize and analyze the chromosomes in a cell, and it is typically done by taking a sample of cells, such as blood cells, from the individual and culturing them in a laboratory setting. These cells are then stained with a special dye that allows the chromosomes to be seen under a microscope, and once the chromosomes are visible, they can be analyzed and arranged in a specific order according to their size, shape, and banding pattern, which pairs them up. And this creates a visual representation of the chromosomes called a karyotype. And karyotypes can be used to identify any chromosomal abnormalities or genetic disorders such as Down syndrome that may be present, because we will notice that in the 21st chromosome, there are 3 identical chromosomes, or not identical, but 3 pair. For all the other chromosomes, there's a pair of chromosomes, while for the 21st chromosome, there's 3 chromosomes, so essentially a trisomy.
Karyographs, also known as ideograms, are graphical representations of a karyotype. They typically show the chromosomes arranged in a specific order and labeled with their respective chromosome number and banding pattern. Karyographs can be used to identify any abnormalities or structural variations in the chromosomes, such as deletion or duplication. In addition, their diagnostic applications, karyotyping and karyographs are also used in research to study chromosomal variation and genetic disorders. They can provide valuable insight into genetic makeup of individuals and populations and help researchers understand the underlying causes of genetic diseases.
The decision to abort a baby with Down syndrome can be a difficult and complex moral dilemma for many individuals and families. And some of the main ethical considerations that are discussed in this context include autonomy and reproductive rights. So, individuals have the right to make decisions about their own bodies and reproductive choices, and this includes the right to decide whether to terminate pregnancy regardless of the reason, because it is that individual's own body. And however, some people argue that this right should be balanced against the potential harm to the fetus and wider social implications of selective abortion.
Disability rights. Some advocates for disability rights argue that aborting fetuses with Down syndrome sends a message that people with disabilities are less valuable and less deserving of life than others. They argue that individuals with Down syndrome can lead fulfilling lives and that society should provide them with the support and resources they need to thrive. And medicalization of disability, or the idea that disabilities are medical problems that need to be cured or eliminated, is a controversial issue. Some argue that selective abortion reinforces this idea and perpetuates the stigmatization of people with disabilities. Others argue that medical advances such as prenatal testing and selective abortion provides women with the information and options they need to make informed decisions about their own pregnancies.
Then we can move on to the social and economic factors. The decision to terminate a pregnancy may be influenced by social and economic factors, such as the availability of healthcare, social support, and financial resources to the mother of the child. And some argue that these factors can contribute to unequal access to reproductive healthcare and may disproportionately affect marginalized communities. So in summary, the decision to abort a fetus with Down syndrome is a complex and multifaceted issue that involves a range of ethical and moral considerations. And it is important for individuals and families to have access to accurate information, counseling, and support to make informed decisions about their pregnancies. And it is important for society to promote inclusivity, acceptance, and support for individuals with disabilities and to work towards eliminating the stigmatization and discrimination they often face.
Maternal age is a significant factor in the chance of having a child with chromosomal abnormalities. Chromosomal abnormalities occurs when there's a change in the number or structure of chromosomes, which can cause genetic disorders. The risk of chromosomal abnormalities increase as maternal age increases. This is because women are born with a fixed number of eggs, and as they age, the quality of their eggs decreases. This can lead to errors during cell division and processes that result in chromosomal abnormalities. And here is a graph that shows the chance of having a child with Down syndrome increases as the mother gets older. This means you can blame the woman. That's a joke.
The most well-known chromosomal abnormality associated with maternal age is Down syndrome. Down syndrome occurs when a child has an extra copy of chromosome 21, as we have discussed in length and detail, and it is more common in babies born to women who are over 35 years old. According to the Center for Disease and Control Prevention, CDC, the risk of having a baby with Down syndrome is about 1 in 1,000 for women who are 30 years old, and it increases to about 1 in 100 for women who are 40 years old.
So, other chromosomal abnormalities that are more common in babies born to older women include trisomy 13, trisomy 18, which are both associated with severe developmental delays and health problems. It is important to note while the risk of chromosomal abnormalities increase with maternal age, especially for women over the age of 35, but the majority of babies born are still healthy. Prenatal testing, such as amniocentesis or chronic villus sampling, can help detect chromosomal abnormalities early on in the pregnancy and allow parents to make their own decision about their pregnancy and care of their child.